Okay, hi everyone. My name is Danielle and I'm a fifth-year PhD candidate in the knee lab. I won't be talking about hemostasis and thrombosis, but I'm going to be talking about the upstream factor that regulates platelets and all our lovely blood cells that bring us here today, known as thrombopoietin, and the role of our liver Kupffer cells. And before I begin, I'd like to say thanks to Dr. Ed Conway and the committee for this opportunity. We'll get right into it. So as I'm sure all of you know, thrombopoietin is the hormone that's indispensable for megakaryocyte differentiation and therefore platelet production, among a number of other blood cells. Briefly, prevailing theory was that TPO acts on the bone marrow to differentiate these platelets, and then platelets in bloodstream bind TPO, resulting in an inverse relationship. However, in 2016, our lab shown that, in fact, GP1B on platelets actually actively regulate TPO at the site of production in the liver. We show that when you knock out GP1B alpha on platelets, you have significantly decreased TPO expression in the liver, as well as circulating protein levels. However, a question that's remained since the publication of this paper is, how do platelets physically contact hepatocytes to mediate TPO? Based on liver architecture, platelets circulating through liver vasculature are physically separated from hepatocytes by a fenestrated barrier that they're just too large to cross, meaning there must be some sort of intermediary facilitating the platelet contact with hepatocytes for subsequent TPO generation. In parallel to this, there's been emerging studies showing that Kupffer cells are indispensable for platelet clearance, leading us to hypothesize that maybe Kupffer cells are facilitating some sort of interaction where platelets contact hepatocytes for TPO generation. So to initially test this, I utilized clodronate liposome and depleted macrophages globally, and therefore also in the liver, and transfused platelets to wild-type and depleted mice. Strikingly, we found that unlike wild-type controls that have significantly increased TPO upon platelet transfusion, macrophage and therefore Kupffer cell depletion significantly attenuates this increase, suggesting they're required. For the sake of time, we also repeated this in a specific transgenic depletion model where only Kupffer cells were depleted, and we observed consistent phenomenon. However, I've left that out again for the sake of time. So we know Kupffer cells are required. However, again, the question remained is, how do they facilitate this mechanism? And we postulated it would either be through facilitating direct platelet hepatocyte contact or some sort of insoluble factor release. And in line with theory one, hepatocyte protrusions have been reported to exist in the endothelial lumen, leading us to wonder if Kupffer cells are facilitating this platelet hepatocyte protrusion contact. So to test this, we utilized a slightly unconventional, albeit rigorously tested model of arsenite treatment, where when you treat mice with significantly low dose of arsenite for a prolonged period of time, they experience significantly decreased fenestration and therefore opportunity for hepatocyte protrusions. Of course, we quantified this and completed a number of controls because, as many of you know, arsenite is cytotoxic, just to ensure there were no confounding factors, and again, not included for the sake of time. Interestingly, we observed that as defenestration occurred and therefore as there was decreased hepatocyte protrusion access to circulating platelets, we also had significantly decreased TPO levels and platelet counts, and these platelet counts intuitively make sense because if TPO is decreased, therefore platelets should be decreased. We then went on to complete an array of imaging studies. Here I have shown transmission electron microscopy, and we analyzed that how platelets were colocalizing with these hepatocyte protrusions and Kupffer cells. And strikingly, in all of our wild-type images, platelets were localized to Kupffer cell surface, and all these adherent platelets were also in direct contact with hepatocyte protrusions, whereas Kupffer cell depleted livers had essentially no platelets in direct contact with hepatocytes. Again, not included for the sake of time, but this was further confirmed via immunofluorescence imaging. And interestingly, when we conducted phagocytosis assays, we observed that these liver Kupffer cells are significantly slower at phagocytosing platelets compared to splenic macrophages and other macrophages in the body, suggesting they may have evolved some sort of mechanism that prior to platelet clearance, exposure on the surface is prolonged to increase hepatocyte contact for subsequent TPO generation. So in summary, this is a small amount of data to an evolving story we currently are hoping to wrap up, where we have identified a central role of Kupffer cells in facilitating platelet hepatocyte contact for subsequent TPO generation. This has important implications in linking platelet clearance to thrombopoiesis and could allow for potentially refined TPO agonist therapies. So I'd like to thank everyone on this slide, and with that, I could now take any questions.
Questions?
Hi, a very nice talk. I have a small question. How did you just delete the Kupffer cell in the liver, and how can you confirm that?
Yeah, we used a Cree-Lox transgenic model that's specific to a Kupffer cell-specific reporter. It's a mouse model where the Kupffer cells express diphtheria toxin receptor under Kupffer cell-specific promoter. So then we dose those mice with diphtheria toxin, and only Kupffer cells are selectively killed, and then just confirm via flow cytometry and immunofluorescence.
So this is nice talk. I'm wondering if you're considering doing maybe some proteomics or something like RNA-seq on the hepatocytes with or without the Kupffer cell knockdown to see if maybe there's something that gets packaged from the platelets through the Kupffer cell or something else.
Yeah, certainly like a very good question. It's something we've talked to collaborators about, so we're considering it.
Good luck.
Thanks.